Okay, thanks. It's really a pleasure to be here. You can hear me, right? Yeah. So this is a survey lecture. I'm not even going to present much of the really most recent something. Anyway, I closed my lab and now I'm running this cancer institute. So I'm not even producing much new, but I do follow everything. So happy to answer questions. So I'm going to address the methods and basic principles of chromatin organization, and you're not going to see one Hi-C map. Okay, so this is for like comic diversion here. Whoops, that's the pointer. Okay, so there are three parts. One is on epigenetic marks, the importance of epigenetics, histone marks in nuclear organization or chromatin organization. The second is about sequence-specific interactions with fixed structures that lead to nuclear organization. And the last is changes during stress DNA repair, which fits the session. So first of all, a long time ago, people used to try to learn about cell biology with EM, and really you couldn't tell much about the nucleus. Even at the highest resolution, you could tell there was a double membrane and there were denser parts and lighter parts, but it was really nondescript. And even when you stain negatively or uracil acetate, all you could see is that there were darker regions and lighter regions that correlated with heterochromatin and euchromatin. Actually, that was more or less the definition. So, but things became much clearer with new techniques or with other techniques. And I really like this electron spectroscopic imaging of David Bassett-Jones, which is very old, but actually he pursued this and optimized this over many years, and it's really a shame people don't do more of it. It gave you a quantitative account of the phosphate nitrogen in an EM, on the level of EM micrographs. And from this, so this is looking at the nitrogen, so protein and nucleic acid, but largely protein. You see the nucleolus, you see dense regions around the nuclear envelope. This is looking at the phosphate, which is much more the DNA or RNA, any kind of nucleic acid, and this is the merger. And you can really see that there is a much more density or concentration of nucleic acid actually at the nuclear envelope and around the periphery. Now then he coupled that with CLEM, which would be using correlative light with EM with antibody specific for covalent histone marks. And then you could see that these yellow regions were actually enriched for K9-trimethyl and K27-trimethyl, representing repressive domains. So it's what everybody thought, but it gave a quantitative vision of this, and it was about tenfold more dense nucleic acid in the compacted regions than in the open regions. So that's history. But the point I want to make is that from yeast to man, chromatin modifications and repressive compartments are one of the major themes. In other words, in yeast, telomeric clusters are repressed and at the nuclear periphery. HP1 at fly pericentric satellite form clusters. This is what I just showed you, again, like that, and polycomb buddies. Now, the point is that these all correlate with histone modifications and types of chromatin. So, in fact, they're listed here, each one a different one. And many of them are species-specific in some ways, because actually the pericentric satellite in fly is not that way in many other species, but it is clear that in this case, it's H3K9 and HP1 that cause this clustering. And the take-home is, and I will give you the backup of this. This is just so you know what's coming. That epigenetic, whoops, sorry, wrong one. Epigenetic marks do not only correlate with spatial organization, they drive it. Now that's a major difference, okay? See, everything I showed you so far was just correlation. You can see correlations all over. That doesn't mean that they're necessary, that has anything to do with driving it nor being first. I mean that you would first have the modification and then the localization. Now, it is important to remember that nuclear organization changes with differentiation, and ES cells, again, look almost like the electron micrographs, rather nondescript, except sometimes nucleally, sometimes around the periphery you see something. But as cells commit to progenitors and then terminally differentiate, you see this enhancement of the separation of U and heterochromatin, and these are, of course, all stages. Since this is the same genome, it's just epigenetic marks that are maintaining the differentiation. And once again, mostly the dense staining heterochromatins either around the nucleolus or around the periphery. Occasionally you see internal clusters. This one's probably around a nucleolus. And then there are always exceptions, and exceptions are important to understand because they put the boundary conditions. And rod nuclei, retinal rod cells have all the heterochromatin clustered in the middle, surrounded by K27. So it's not really the canonical organization. And of course, senescent cells, as shown by one of Wendy Bickmore's postdocs, is also full of these heterochromatic foci. And they also have the same organization where you have a core of K9 surrounded by K27 methyl and then other marks. So special cases happen. That just goes to say that nuclear organization is definitely not fixed. It changes with differentiation, it changes with cell state. So what are the sequences that are at the periphery? And one of the techniques that I think was very instrumental was developed by Bas van Steensel. It's where you tether bacterial methylase to any protein you want to localize, but in this case, it was tethered to lamin or emerin in order to identify the sequences that were close in proximity, because if you methylate the GATCs, they become cleavable by a certain restriction enzyme, and that tells you which sequences are located there. So this was done genome-wide. I think this is textbook material. Basically, the heterochromatin at the periphery turned out to be, of course, enriched for the lamin DAM-ID, and genes tended to be depleted for lamin DAM-ID, and so it just reinforced what everybody thought. Now, Bas did this as well in a 2010 paper looking at ES cells and then differentiated neuronal cells, neuron-like cells, and could look at specific regions of specific genes. And what he found was that indeed in the ES cell you might have a whole region where silent neuronal specific genes would still be at the nuclear periphery, but stem cell genes might be extended out away from the periphery. And then when you differentiated, these neuronal genes would be active, and then they would now no longer be as closely associated with the lamin. So that it suggested that actually lamin binding was not just somehow sequence specific due to repeats, but that genes actually associate with the lamin when they're repressed in a tissue-specific manner. Now, this was reinforced. Wendy's lab did some of this, but also there were other labs that did it even before, where they look here at a mammalian nucleus, a keratinocyte, look at its differentiation in the mouse, in actual mouse differentiation, and you see that the chromosome territories are in red. The specific cluster of genes is in green. And what you see is that at early development, when the cells are not activated to become keratinocytes, they're more at the periphery, and then as you go through this differentiation, you see that they move inside, but the the chromosome territory doesn't move. The whole chromosome doesn't move, just the domain. So this was done by fish, because that's the way people did it back then. And then we did something very similar with live imaging in C. elegans, and we would tag the promoters of tissue-specific genes, a number of them, in undifferentiated embryonic nuclei up to gastrulation in C. elegans. There was no specific or enrichment of any of these tissue-specific promoters, either at the periphery or in the inside. But in differentiated tissues, then we would see the active promoters would be, without fail, recruited to the internal portion of the nucleus, and the tissue-specific promoters that should be repressed were sequestered at the periphery. So this was, for me, extremely clean data in C. elegans, because we could look in vivo in a real organism differentiating. And the only thing that was maybe a surprise was that housekeeping genes just didn't care. So whereas tissue-specific genes seem to have a promoter that would recognize that it should be either on, if it were off, it would be at the periphery, and on, it would be internal. Housekeeping genes could be on anywhere. So the take-home is that, yes, there's a differentiation-specific localization, but it's very much dependent on what kind of promoter. So how general is this? So the correlation in worms is not only a few promoters off and on, but actually the whole chromosome. This is chromosome 1, but all worm chromosomes are the same. The ends of the chromosomes have a lot of repeats. So this is looking at H3K9 mono, di, and tri. And actually, I haven't mapped here the repeats, but the repeats actually correlate almost exactly with that. The repetitive regions, the transposons, simple repeats, tend to be more enriched in the arms. Genes are more enriched in the middle. And if you map it relative, this is by chip, not by DAM-ID, but we've done DAM-ID, it gives the same result. You see that the extremities of the chromosomes are bound to the nuclear envelope, and the centers of the chromosomes tend to stick in, so you have sort of this kind of arrangement of chromosomes. Now, it's a nice correlation, and we could confirm all this, but it doesn't tell you if it's causal. And what I was telling you before, correlation is not causation, and if you really want to understand whether anything's important, you have to do genetics. So the proof of causality must be genetic. Actually, Angela and I wrote a review in 2004 and basically said that. I don't know if anybody read it, but it's still true. So what we did was create an artificial array of heterochromatin for the worm. It's integrated in a chromosome, and it's a tandem array about 250 times of single lacoocyte, so it's not an array of lacoocytes, but it's a tandem insertion, which worms are very good at doing, that has a couple, like a duplex laco binding site. And it expresses GFP lac-I, which then binds to its own lac operator, so that you have a feedback system where at low-level expression, you see these two spots. This is an embryo. Each one of these is a nucleus, and each nucleus has two spots. It's diploid, obviously. And you see that they're all at the nuclear periphery. So how do we know that it's at the periphery? We always quantify. This was sort of like the obligatory method of the lab, was that if you look at anything, you have to quantify it. And fortunately, all the nuclei we worked with, yeast and worm nuclei, were spherical. So that simplifies life a lot. If you have a spherical nucleus, you can do a through-focal stack. Take the plane where the spot is the brightest, it doesn't matter where it is, and divide that plane into 3 zones of equal surface. Now, there's a biophysical law that says that you don't have to do it in 3D. That will be equivalent to measurements in 3D if you count enough of them. And of course, the array was enriched at the zone 1, which is the peripheral surface zone, and it correlated with all the repressive marks. So Ben Taubin, years ago, decided to do a genome-wide screen to identify factors involved in heterochromatin anchoring so that we could then knock them out and ask the question, is it causal? Whatever it is. And so at low-level expression, these are worms, so you don't see any GFP much. I mean, it's a very low level. And if it's de-repressed, then you see very, very bright expression of this LAC-I GFP binding its own array. So he did this genome-wide, 20,000 genes in triplicate, and he did the whole screen more than once. There were about 29 regulators that de-repressed heterochromatin in worms. Most of them were heterochromatin marks, the ones you expect, K9-methyl, K27-methyl, and some that we didn't expect. But most of them fell into the category of explicable chromatin marks from everything we know, a DNA degradation machinery that was studied further. But when we asked the question, out of all these hits, which ones caused the delocalization, not just the de-repression, there was only one hit, or actually it was two hits, but the same enzyme, S-adenosylmethionine synthase, and this caused both de-repression and delocalization. Now, the explanation of that is that the donor for methylation is SAM, and it requires SAM synthetase to be created. And so it's the donor for all methylation. So we knocked out pairwise, dozens of histomethyltransferases, and two of them, when they were knocked out together, caused delocalization and de-repression of these heterochromatic repeats that were at the embryonic nucleus periphery. So a MET set, this is the loss of two H3K9 HMTs. The array shifts inside. If you knock out just one or the other, you de-repress, but you still stay anchored, partial de-repression. And in wild type, of course, you could see that the K9 trimethyl was completely at the periphery. So K9 di and tri is important both for repression and position, and this was that paper. Now, and interestingly, the methyltransferases stay bound to the repressed array. So that was also a nice, you don't see the other genomic loci, but there are genomic loci, but this is such a sink for this methyltransferase that it stays there. So this was the summary, and it is important to know because this is the pathway to proving that there's a function for epigenetic marks in nuclear organization. And that was showing that the monodimethyltransferase for K9, it's specific for K9, and this MET2 is a SETDB1 homolog, the mammalian homolog, deposits mono and di, and this is sufficient to be sequestered at the nuclear envelope, and the SET25 adds the trimethyl, which then leads to full repression. So that's nice. We showed that there's an epigenetic mark that determines position and repression, but is it causal? If it's causal, then we should be able to knock it out selectively. That would be the dream, to selectively knock out either position or the mark or repression and see if they're the same. So we did another screen, and this was for anything that would read the K9 monodi and trimethyl mark, because if you had an anchor, if it were directly mediating position, then you should be able to find the protein that is reading that mark and is at the nuclear envelope. So this was the work of Adriana Gonzalez in the lab. It wasn't a whole genome screen. We screened, because in this case we had to measure distance from the periphery for every knockout. It was quite laborious, but she had a great hit. It's called chromodomain protein 4, so it obviously has a chromodomain which recognizes K9 methylation, and a large IDR, so denatured, or a non-structured domain. So the chromodomain is very similar to the HP1 chromodomain, and a less structured domain is the C-terminus. You need both sides of it to mediate function. And if you just take this chromodomain and ask what's its affinity for different, we tried every histone mark we could, it actually was extremely selective for K9, mono, di, and tri. There was one other mark it recognized, which was K37, which actually doesn't appear very frequently. And so this is measuring KD, and the lower the KD, of course, the higher the affinity. And it bind mono, di, and tri, even though mono K9 methyl is not repressive. So, and this is the peptide without any methylation. So then the best side of this story was that when we labeled it with RFP, it was clearly at the nuclear envelope all by itself, whether you had K9 methylation or not. So this itself is localized at the periphery. And if we made a point mutant in the chromodomain so that it could not bind the K9 methyl or knock it out completely, we completely delocalized the heterochromatin array from the periphery to the inside. And the CEC4 stayed peripheral. So this is the quantitation. It's clearly random, and this is prior. So now we really have a story. K9 methyl not only can mediate the association with the membrane, it does so through a reader called CEC4, which anchors but does not silence. I'll give you that data in a second. And then the trimethyl, or the di and trimethyl actually bind the HP1 or a second Lin61 reader, and they silence but do not anchor. And if you knock out CEC4, and leave the MET2 functional MET2 set25 and the readers, you can silence without being at the periphery, at least in embryos. So this is really the impact how a single histone modification can drive nuclear organization of a whole set of chromatin domains. So as I said, if you knock out CEC4, leaving the HMTs and the HP1 intact, you allow array repression away from the nuclear envelope. Now, what we found then was that was all done in embryos. The nuclei were nice and spherical. Everything could be quantified. But as we looked at tissue differentiation in the CEC4 delta, they did have a bit of problem differentiating in robustly, but the array did not leave the periphery in the adults. So it was really funny. The array would be internal in the embryos, and as different cells differentiated into muscle, gut, and neurons, the array went back to the periphery. So this was really, this is amazing, because you knock out the anchor and there's still another anchor. So this was clear. There had to be another second anchor. And we wondered if it was cell type specific, what it happened in every tissue we looked at, and whether there are general rules. So this is just one slide to summarize a very complex story, but I think it's important. As I said, in embryonic cells, the CEC4 recognizes the K9 methyl and sequesters it clean, right? And in differentiated cells, wild type, as I said, you can knock out the CEC4 and the array at least, and much of the heterochromatin stays peripheral. So we assumed there must be a second anchor. We didn't know what it was. So we did a screen, and this was a genome-wide screen, or almost genome-wide. And what we found was that when we knocked out MRG1, which is actually a reader of an active mark, we interfered with the second anchor, and the heterochromatin finally left the periphery. So it was really confusing. But we found that MRG1 actually binds the active mark H3K36, and actually we could knock out H3K36 and have the same methylation and have the same. And when it is lost, then we antagonize this second site. You have to actually knock out CEC4 as well. And we found that MRG1, actually its role is to sequester, we're not sure if it's direct or indirect, CBP, which is a histone, the major P300 histone acetyltransferase, away from heterochromatin, or promoters that would have CBP type binding sites in heterochromatin. So, and what happens when you eliminate MRG1 is that actually the CBP1 invades heterochromatin and then antagonizes the second promoter, and that what we saw was that there was an accumulation of K27 acetyl, as you find at active promoters or at enhancers. So what this said was that actually there's an antagonizing factor for this second anchor that is probably a promoter acting on promoters, and will antagonize whatever this other ligand site is. It means that in differentiated cells, although this pathway is still functional, there's a second pathway that sequesters inactive promoters, and there is a pathway that sequesters CBP1 or CBP or P300 away. So this is actually another important finding, that the compartments are not only silencing compartments, but active compartments actually have a, if you will, a sequestrating force themselves. And you who study, enhancers and promoters interaction, of course, that must ring a bell. But interestingly, it's very important to keep those activating enzymes sequestered away from the periphery in order for silent chromatin to be tethered. So how does this play out in mammalian cells? I only have one slide because it's complicated. So mammalian cells have the lamins, and they have many of the same conserved inner membrane protein molecules that extend. Some of them have interaction with chromatin indirectly. They do not have a CEC4 homologue, although PRR14 is close, but it's only a tissue-specific type interaction with heterochromatin. It seems to be more active in differentiated than in early embryos. Now, what we do know from Irina Solovay is that if you knock out the lamin B receptor and you knock out lamin A, you do delocalize heterochromatin, but they don't know the interaction directly or indirectly with chromatin, at least. Probably it's through a reader of either K20, H4K20 or K9 methyl, but it's not clear. There's another anchorage site called the Cache Sun Domain Complex link, which also reaches in and binds chromatin, and this is also conserved from yeast to man. So actually we don't know much more, and I'm not going to dwell on it, but this is a fruitful field to study, and for sure there are going to be interaction sites, and my guess is that in differentiated cells, a lot of it will be through transcription factors that bind these various proteins and sequester domains away. So that's all I wanted to say about epigenetic marks. Now, the other two are much shorter. But the take-home is that an epigenetic mark can completely actually mediate sequestration of domains. It does so through readers, and it also mediates repression through readers. They are not coincidence. They are not, at least in worms, they do not have to be identical, and actually we know that in mammalian cells as well, and in yeast. But epigenetic marks are a major player in chromatin organization, I think, but you have to prove it genetically and not correlative. So the second little vignette, and this is very short, is about sequence-specific interactions. Everything I've told you now is about a mark on a histone. That means that it could be anywhere in the genome, and it would do its job. Now we're talking about sequence-specific interactions with fixed structures, once again, mostly at the nuclear envelope, because and I'm only giving you a personal view of this. It's one example, and it's the example from my lab. So years and years and years ago, we studied the clustering of telomeres. The yeast has 32 telomeres in a haploid cell, 64 in a diploid, and they cluster together in these foci, either at the edge of the nuclear envelope, but the nucleolus, or at the nuclear envelope. This is staining for nucleolar. This is anti-RAP1, the telomere repeat binding protein, and this is just DNA. So How did we know from pictures like this that telomeres were clustered? Well, we did do fish, but fish is not very efficient. But we did fish on meiotic spreads and could show that this RAP1 antibody colocalized exactly at yeast telomeres. It also binds, actually, repressor elements and promoters, but its most abundant binding sites are at telomeres, and you see it's on this was actually the first proof that RAP1 bound telomeres. It was years ago. So we were looking at this in the context of silencing, because subtelomeric regions are silenced by the SIR complex. And indeed, SIR proteins made the same kind of spotty distribution at the nuclear envelope. We did SIR protein colocalization with telomeres in situ, and lots of complicated things, because that's what we could do in the 1990s. And we found that there were conditions that partially de-repressed telomeric silencing that also dispersed the SIRs. Now the question that arises is, what is the mechanism, right? Is it telomeres that have some affinity for the periphery, or is it silent chromatin that has if it's just silent chromatin, then it's a repeat of just what I told you about epigenetic marks, although the mark in this case is an absence of acetylation. And the story is that it's actually both. Telomeres bind RAP1, but they also bind an N complex, which involves Cu. And between the RAP1 binding the telomeric repeat, so that's a sequence-specific interaction, and Cu binding the end, they tether the end of the chromosome to the nuclear envelope completely independently of chromatin. So you don't need any nucleosomes, you don't need silencing, you just need RAP1 binding on multiple sites. It binds SIR4, which is one of the silencing proteins, but SIR4 doesn't silence alone, it absolutely requires SIR3. And SIR4 interacts with a protein, and this is again Angela's work, called ESC1, which is a transmembrane or a isoprenylated protein, and MPS3 binds Cu, and EST1, which is a subunit of telomerase, and angers it at the periphery. So this actually happens independent of nucleosomes. We've knocked out, this was all reconstituted over years of study. But there's also the silent chromatin pathway. So dependent on silent chromatin, you also bind the SIR complex, which is what actually silences the SIR3 binding the face of the nucleosomes, protecting it from polymerase and other nucleolytic attack. SIR3 binds SIR4, SIR4 binds SIR2, which allows propagation, and then the same mechanisms of anchorage kick in, plus an association with nuclear pore, which I always was never convinced about this, and yet some very recent papers completely convinced me that this is actually, this NUP170 forms a complex with Cis2, which is a sumo ligase, and ESC1, and SIR4. You can recover this complex, and it clearly is involved in the tethering of silent chromatin and possibly also telomeres alone. So there are three pathways. There's the sun domain protein. This MPS3 is the sun domain homolog, which I said was conserved. There's the isoprenylated nuclear envelope protein like ESC1, and then there's a pore protein that actually not only binds pores, it's a partial subcomplex of the pore that is also found at other sites along the nuclear envelope. And these are papers that are at the bottom here. So one pathway is dependent on silent chromatin, the other is dependent on sequence, which is the telomeric repeat or the end of the chromosome. So in fact, you can tether, you can organize chromosomes, you can actually specify subnuclear organization purely on a sequence motif. Now, deletion of any of these anchor proteins as well as the sumo-transferase Cis2, it turns out that sumolation is important in many of these intermolecular interactions, can partially de-repress TPE. So it also has a functional role. So the last bit on this section about, so telomeres go to the periphery, they subtelomeric regions accumulate SIRS. The whole question is, why does this? Why do we see that the anchors, knocking out the anchors, affects silencing even partially? And now I'm going to take you back even further to the mid-90s. And this is a paper I published with Eric Gilson, who was a postdoc in the lab, because we had noticed that it mattered. This was our reporter construct where you put two silencers that had RAP1 sites and other sites that would nucleate SIRS. And you put it different places on the chromosome, and it didn't work. So if it was near the telomeres, it did, which was its natural location, and anywhere else it didn't. So we asked the question, why? You know, it fit with the idea that actually the telomeres were anchored and presumably the internal parts were not. It didn't tell us why the internal parts wouldn't just go bind, but one idea was the silencers, which should nucleate the SIRS, didn't work when they were too far away, when there weren't enough SIRS, and the telomeres, as we had seen, sequester the SIRS. So we overexpressed on SEN plasmids, so this is just one extra copy of SIR3, one extra copy of SIR4, both of them together, and now we could silence internally. Or we could insert a cluster of TG repeats, which would bind RAP1 and bring in SIRS, and that would also silence. So actually, this works. I mean, what this telomeric clustering does is actually concentrate SIRS in a domain so that the silencers, the nucleation of silencing, can work. So it's very old stuff, but I realize that not everybody understands that a twofold difference in silencing protein can make all the difference to the status of chromatin. And why is that? Well, it's because repeats and silent domains, and anyway, any kind of clustered sequence will create like a sink for certain proteins. And in fact, you can convert in repressive chromatin, a linear gradient from concentrated to less concentrated can be converted into an on-off state, so silenced and not silenced, simply by creating a kind of threshold for switching into a silent state. And that's exactly what we see. And when you have a gene near a telomere, if it's close enough, it'll be silenced. As you go further away, it won't be. But it's not like you go gradually into the, you know, the sunset. It's like it's daylight or it's night. And that's very important to remember. It's an on-off switch when you concentrate key factors. Whether that's LPS or not, I'm not going to discuss. I'm not going to discuss anything that's controversial today unless you ask me. So, and this was, again, Angela's work. So the question was, if you tether anything at the periphery near these foci, it will be silenced. That was shown by Androulis and Sternglanz in 19-something. But a very important experiment was to ask, is it the nuclear envelope or is it really the clusters? So we did it in a SIR mutant where the SIRs are, not a SIR mutant, a COO mutant where the SIRs are dispersed. And the tethered URA gene was on rather than off. But the interesting thing was done by Angela, and that was to look at an internal reporter, or look at internal genes, actually, and could see that actually if you had strong enough nucleation sites, you would silence internally and not at the periphery if the SIRs were no longer sequestered. So proper segregation into these, let's say, clusters, these foci, or if you want condensates, but I don't think they're condensates, is crucial for maintaining both the on and the off state. So that was the foray into the past. Now, the last 2 minutes, I'm going to tell you about the changes during stress, and then you're going to hear it from 3 more excellent talks. So I'm not going to tell you everything I know about DNA damage, even though I worked on it for years. But everything I told you about segregation, chromatin marks, changes when you stress the cell. So if you induce either nutrient stress or starvation or heat stress or DNA damage, the SIRS, and this was again a 1990s paper. I'm sorry to impose all this old stuff on you, but I'm sure you didn't read it. The SIRS become dispersed and somewhat RAP1 in the face of DNA damage. Now, if you block the checkpoint, it doesn't become dispersed. So that was interesting. And if you asked what happens to telos, the telos stay anchored, but they become partially, not completely, just partially, maybe tenfold de-repressed. So at this time, we were studying all kinds of single molecule behaviors, or single site behaviors by tagging loci in yeast and tracking them in real time. And we used markers for the pore, and I'm sure many of you have seen this kind of movie where each one of these spots is a single locus, in this case 2 telomeres, in this case 2 telomeres, and we could ask how frequently different telomeres touch each other and so forth. But what we really wanted to do was learn what we could from this interesting movement of DNA. And so we optimized. This was, again, about the same time that Sadat had published the first looking at tagged regions. We coupled it with the nuclear pore so we could actually quantify movement more precisely. And we would normalize movement, so eliminate all transversal movement by subtracting the movement of the nuclear pore, the whole ring, from the spot. Anyway, that gave us MSD plots of every interval of imaging. At the time, we were doing 1.5 second intervals. Now we do about 80 milliseconds. And you could plot the mean squared displacement. I'm sure you all know about this, and ask the character of the behavior of the movement. Now these two guys, this fellow was at MIT afterwards. He had come from Israel and worked with Holtzman in Paris, and my graduate student, Andrew, really worked very hard together to try to get a biophysical view of what this movement meant. Asaf convinced us that we had to do much, much more frequent imaging, so we were doing 80 milliseconds, sometimes 300 millisecond intervals instead of 1.5 seconds, and then we could, of course, for shorter time frames, and then we could extract biophysical parameters of confinement, tethering interactions, diffusion coefficient, and anomalous exponent. So I'm sure, since you're physicists, some of you, this is really boring, but the scaling exponents describe single particle movement. As you know, Brownian gives you an alpha, a derivative of 1, and greater than alpha is directed motion and less is constrained, and all the movement we were looking at was constrained subdiffusive near random walk. Now, we would use Asaf's methods, they're all published, or formulas to calculate the various parameters. And we could model then what we were looking at. And the modeling argued to us under certain conditions we could see alpha increasing slightly, but from all the other parameters, what he suspected was that the kind of constraint we were seeing on movement could actually arise simply from nucleosome-nucleosome interaction. Of course, I love this idea because I love nucleosomes, I love the histone modifications, and that means that histones might drive this movement. In fact, it was also relevant to DNA damage because Vincent Dion and Judith, when she was with Rothstein, had shown that actually a double-strand break, if it's tagged, will increase its mobility. It'll still be a constrained random walk, but it will increase its mobility almost threefold in the volume of the nucleus. We asked the question, why? And we're, again, I'm not saying there's no directed motion, but at the resolution we're looking at things, all we could see was random motion. And Asaf proposed that actually it could be the packing of nucleosomes itself that was changing. So graduate student Mickey Hauer looked at nucleosomes, and what he found was not that it was just modifications of histones, but actually the level of histones dropped about 20% of all 4 core histones, but not HTZ1, and it was random, it was across the whole genome. So in recent times then, we tried to map, what is the mechanism of this, because we wanted to get at the mechanism to know if it had an impact on repair. And we could find that there were 4 ubiquitin ligases that actually target histones in different ways. Some of them were known, others were not known to be nuclear histone mediators. They were all downstream of the checkpoint response, so you have to have a significant level of damage. But then what happens is about 1 in 5 nucleosomes is degraded. Now, the interesting thing is that does affect then the homology search, and that's what you'll hear about from others later. So that's the summary. Epigenetic marks can drive nuclear organization, so pay attention to your nucleosomes. Sequence-dependent positioning occurs occasionally, but usually with very specific sequences, like telomeres or special sequences. And chromatin responds strongly to stress and DNA damage and can even affect the density of histones. Now, there are many scales to study nuclear organization and chromatin organization in the nucleus. And as you have noticed, I only talked about this. That's because I worked on yeast and worms. You can do TADs in yeast and worms, but for the things I was doing, it was mostly single particle or positioning. I did talk about A and B compartments, B being heterochromatin, A being, or peripheral, and A being active. But I understand that the rest of your whole week has been talking about all the other stuff. So I don't apologize. This was a foray into other methods. So this was my lab at the 2015. We meet every 5 years, so we'll meet again next year. And there are too many people to thank, so I won't. I had a great lab and had a lot of fun. Thank you. And I'm happy to take questions. 10 minutes, I guess.
Thank you, Suzanne. So, questions? Aurèle.
Hi, Suzanne. So you showed this mobility of the chromatin near the break site that you characterize alongside rastin, and in one of your latest paper, you show that you have one mutant, I think it's a ULS1 mutant, that is completely defective for the mobility of the break.
But you still recombine.
Come again?
And you still
recombine perfectly well with a donor located somewhere else in the genome.
Right. And that because the histone degradation still happens. So we think it's access. I mean, you have to have some mobility, but the basal level mobility might be enough. I mean, there's always mobility, and we were seeing enhanced mobility. So the question is, do you need that enhanced mobility to do the search? What I think, this is based on our results, is that you may not need the enhanced mobility. What you need is accessibility of the long RAD 51 needle or whatever. It still has to find the homologous sequence. And if you have nucleosomes packed at the normal density, it will happen at a certain rate. If you have so much damage that you activate the checkpoint, then you're going to get rid, at least in our hands, you get rid of 1 in 5 nucleosomes, which of course enhances the frequency. So this was Sheblal et al. I thought it was a great way to end your career on that topic, because actually it was counteracting maybe our paradigm that we thought for maybe 10 years. That, oh, this must homology. If you do modeling, movement, of course, has an effect. The more you move, the more, and we did modeling with, the break site tethered, or just the target site tethered, or anything you want. We did Monte Carlo modeling, we did everything, Brownian. But I think it's probably accessibility is more rate limiting than contact. Now, there is loosening up, even if you don't see a very big increase in mobility. What we did see is that along with histone degradation, there is some degradation of MRX, okay, because you have to free up the end, otherwise MRX is going to hold the two ends together, and of cohesin. Cohesin is loaded, but then shifts along the chromosome arm. So actually, we also found effects of knocking down cohesin and MRX. So that's my only one caveat about that simple interpretation that it's accessibility, is that if you prevent release from MRX and cohesin, you also limit somewhat the recombination, the strand invasion. But as you know, I would have loved to use your method, and we'll hear about it in an hour or so, because it might have given us more accurate information than the assay we used, which was actually depending on complete strand invasion and then PCR. So it was looking at the end product and not the actual invasion event. But it was the best we could do at the time, and that's the answer.
Thank you.
Thank you. Other questions?
Hello, thank you. Very nice talk. I have a curiosity. You mentioned stress, and then you talk about DNA damage. Did you try other kind of stresses, such as
Have such a strong, well, obviously DNA damage has the strongest, and it is, at least in our hands, dependent on activating the checkpoint kinase. Almost all the changes we see, if you knock out MEK1 or RAD9, you don't see such strong chromatin responses. This is interesting because that means the checkpoint kinases are telling the nucleus we have a problem, you know, and not just activating a pathway of repair. Originally people thought, oh, you know, you turn on the genes you need to repair, but actually it does a lot more. Other kinds of stress. In worms, heat shock has quite an effect on gene position. And Angela studies nutritional stress of various kinds. So I don't want to, I can't generalize about that. Each stress probably has a different kind of effect. But the only one that we really studied, both in worms and yeast, was DNA damage, and this triggered. So it's not low-level DNA damage. It's not any small blip in the DNA. It's checkpoint activation.
Thank you.
So you showed this data that had to do with a certain point moving away from the
periphery, yeah?
Periphery, etc., right? And from there you extracted the strength of the interaction between DNA and the periphery, as well as between nucleosomes.
Yeah, but that was a purified. That KD was not in vivo. The KD was done with purified chroma. Are you talking about the worm?
Yeah, I mean, you showed that movie where things were moving.
Oh, I see.
Yeah.
Oh, okay.
There was the data that I missed it, but you had a table with the very end. Yeah, maybe the one after this.
So this is not, okay, actually double-strand break, I mean, depending on the type of damage, oh, this gets very complicated, okay? Damage that can be repaired, resected, a normal double-strand break that's resected and then has to find a template, these are the conditions under which we see mobility increase, okay? We do see it, well, no, that's not quite true. We also see it with zeaxanthin, which is a general single and double-strand break-inducing agent. But but if you have a blocked double-strand break, where the ends cannot be resected, or a collapsed replication fork, these actually, it's not that they move away from the periphery, they move to the periphery. And that I didn't even enter into that. That's complex, but it has to do with sumylation and the fact that a number of key sumylation enzymes are associated with nuclear pores. And what probably happens is that a blocked, let's say, a double-strand break or any kind of collapsed replication fork where you cannot simply, find a HR and find a homolog, you usually have to process that end, either degrade something that's blocking the end or resect it. So what happens there is that it gets sumylated, it gets transferred to the nuclear pore, where a sumo-dependent ubiquitin ligase called Stubel and the SLX58, RNF4 in mammals, ubiquitinates and then degrades factors that are preventing, let's say, a kind of recombination or illegitimate recombination which would allow repair. So there is no simple conclusion about where damage goes. Okay, it depends absolutely on what the ends, what is wrong with the ends, and what stage of repair you are, and what kind of repair you're undergoing. So the only thing we saw moving away, damage moving away, well, actually, we've never seen damage moving away from the pore except after it's somehow processed through sumylation, ubiquitination, and degradation. So I'm not sure if that answers your question, but position of damage is quite different from just mobility of damage.
Thank you very much for the nice talk. So my question was, do you see also histones degradation in other organisms than yeast? Is it something general?
Yeah, this is a good question. So, so your colleague, Sophie Polo, has shown that actually there's a displacement of histones after UV radiation, and I think she's used other kinds of damage. So actually her idea is that actually chromatin does expand. Same thing, I didn't show you the pictures, but what we observe is that chromatin expands. Not the nucleus, but chromatin loci expand when you have this checkpoint activated histone degradation. And she attributed it to displacement of nucleosomes. If you look carefully at her gels, she sees about 10% less histones, but this is mammalian cells, so it's much harder to see a significant drop than it is in yeast. So my guess is it may not be degradation in other organisms. On the other hand, we also know that every single repair pathway we know uses ubiquitin ligases like crazy, and nobody knows what they do, basically. I mean, a few mediate protein-protein interaction. But my guess is we've ignored maybe some of the transient degradation of histones, or maybe certain modified histones. Maybe it's just that, certain histone variants you have to evict. So if you want to know, exactly how universal that is, I'm not sure. But I am sure that chromatin globally responds to high-level damage. And I think we'll hear that from others.
If there is no further question, I think we should thank Suzanne and have the coffee break.
Okay, thanks, Angela. Thanks to all the organizers for their kind invitation to this nice meeting. So today I'm going to try to introduce a little bit what we know, or at least what we knew about the DSB in the context of chromatin and chromosome architecture and nuclear organization. And then I'm going to talk about our own work. So I'm sure all of you know that DNA double-strand break are really harmful lesions that are rapidly detected and repaired on the genome. And they are handled by various repair pathways, which I will not go into detail. Homologous recombination, I think Aurèle will probably nicely introduce that pathway, and non-homologous enjoining pathway. And what is important is actually all of this repair pathway, they won't trigger the same consequences on the genome, they won't leave the same scar when inefficient, inaccurate. Knowing that homologous recombination will use an intact copy of the locus as a template, so it's usually quite accurate unless the wrong copy is used. Okay, so one of the biggest questions in the field is how does the cell decide between all these pathways to repair each single DNA double-strand break. So we know the cell cycle plays a role, and now for the last 10 years we know that the chromatin environment is a key determinant of these repair pathways. So what do we know about the repair of DSB in the context of chromatin? So as I just said, what we have learned is that, first of all, the pre-existing chromatin context, so which means the genomic location of the break, will contribute to determining the repair pathway, targeting more toward non-homologous enjoining or homologous recombination. And what we know is that transcribe unit will deploy a specific repair pathway. That's also the case for constitutive heterochromatin. So when it is damaged, they will undergo specific repair centromere or facultative heterochromatin. Then, beyond that, what happens when you have a DSB is that it will trigger chromatin changes. So first, and that occur at different scale. So from the really close proximity of the few nucleosomes around the DSBs to more global modification at the Taddei scale, and I'm going to talk about that, to some large chromosome reorganization within the nucleus. So, as I just said, around the DSBs, we know that there is a number of chromatin process that will take place, and that will favor nucleosome removal in order to promote the accessibility of repair factors, the resection process, and the DNA synthesis. And there is a plethora of histone modification, histone modifier, nucleosome remodelers that will act at each DSBs. And what we figured out a few years ago is that we can determine histone modification that will be associated either with non-homologous enjoining or with homologous recombination repair. So there is a kind of specific repair pathway histone code. Then I told you some of this modification can actually spread on really wide domain. That's the case of the very famous gamma H2AX modification, which is the phosphorylation of the H2AX histone variant. And what we could determine by ChIP-ChIP is that that modification is able to spread on 1 to 2 megabase. And that formed these big visible foci in the nucleus. And beyond those 1D change, what we know then is the chromosome are reorganized within the nucleus, chromosome architecture change. So, for instance, so there is plenty of labs that have worked on these issues. So, for instance, DSB can relocate in some specific locations. So, Suzanne discussed a little bit about that in the context of persistent break in yeast. But that's also the case for instance, breaks that are induced in the ribosomal DNA. So that was in human cells. When you induce break in the ribosomal DNA, it will actually be excluded from the nucleolus in order to be repaired at the nucleolar periphery. And this occurs also on human cell, and what we could find is that usually when that happens, so sorry, here you have the DSB that were induced in the ribosomal DNA, and they can actually contact nuclear envelope and vagination. This relocalization also occurs for DSBs that are induced in heterochromatin. So that's the case in mouse cells. So if you induce breaks in heterochromatin, the break will relocate at the periphery of the heterochromatin focus. And in Drosophila cells, it was even shown that that specific break further moved to the nuclear envelope in order to undergo homologous recombination repair. Right, so, and there is another process that has been a lot studied as well, which is what we call the DSB clustering, so-called DSB clustering. So that was very clear in yeast for a long time, that two DSBs can actually cluster together in the nucleus, but in mammalian cell has been controversial for a long time, with some studies, for instance, that study from the lab of Renan Canard back in 2004, that actually could see that when you do laser stripe in G1 in human cells, then you see that along the time, then the DSBs will actually cluster together along that line. While in some other lab, they couldn't actually found post-irradiation the movement of those DSBs and their clustering. So it was clear that something was going on in terms of how you do induce the breaks and what is the kinetic of that clustering and the cell cycle phase, etc. All right, so I would say that's the biggest question that we have in the lab and in the field regarding chromatin and repair. So this is how does repair varies across the genome, how does chromatin actually contributes to address each specific repair pathways, and what are the local and the spatial chromatin and chromosome changes that are induced by DSBs, and how does this change contribute to the efficiency and the accuracy of the repair process. So in order to tackle this question, a few years ago now, we decided to turn to what is very famous in this audience, which is chromosome conformation capture approach. So I will not discuss what it is to this audience, but we decided to do that using a system that we developed now a long time ago, where we can actually induce multiple DSB in the across the human genome at annotated position. So to do that, we use a restriction enzyme that is called SIS1, and that is fused to the ligand binding domain of the estrogen receptor. So when we add the hydroxytamoxifen, the enzyme goes in the nucleus and is able to target about 100, induce 100 DSB on the human genome. And that enzyme is also fused to an auxin-inducible degron, so when we add auxin, degrade the enzyme, and the DSB can get repaired. Okay, so we use basically, because the breaks are annotated, we use this system together with a lot of high-throughput genomic approach in the lab. So for instance, here what you can see is that we use BLESS, which is this experiment, NGS-based experiment that allow you to determine the position of DSBs on the genome. And you can see that when you add hydroxytamoxifen, we have this very sharp peak that pops up at SIS1 position. Okay. So that's basically how it behaves. We have about 1,200 sites annotated on the genome, but we are able to cut only roughly 170 sites on the genome efficiently. Okay, so there is a lot of DSBs of sites that are not accessible with our genome, but what is with our system. But what is interesting is that more than half of those DSBs are actually lying in active loci, while some are lying in inactive and loci and intergenic loci. So we can compare what's going on between all these different positions. Okay, so now let me present you briefly 3 different stories. I'm going to try to be quite short on each. But the first one is about how do you build those gamma H2AX foci, and how do you ensure spreading of gamma H2AX in a really rapid manner from 0 to those big, large domains on the genome. And that was the work of a really talented PhD student who now left the lab, Colin Arnoux. So what we knew from our CHIP-seq experiment is that when you induce DSBs, so it can spread on very asymmetrical pattern. Here, for instance, the DSB is here, and it can actually spread on quite asymmetrical. And it's not only gamma H2AX, it can be ubiquitin chain as well, 53BP1, and the depletion of the histone H1 as well follow the same pattern. So what Colleen decided to do was to look at the chromosome architecture, at that chromosome conformation at those positions. So she decided to perform 4 C-seq experiments that I guess I don't need to also describe on this audience. So basically, you're looking at, you use a bait, so you're looking at all the possible loci that interact with your specific viewpoint. And she used as a viewpoint a DSB that is annotated in our system. And she performed this for C-seq before any break induction, and I hope you can appreciate how similar it is to the gamma H2AX spreading that is after break induction. So basically we knew that gamma H2AX was spreading in a chromosome conformation, chromosome structure. This is the case for gamma H2AX, but when you look at the kinase that is phosphorylating H2AX, so the activated kinase, phospho-ATM, you can see that actually the kinase is not able to spread. So the kinase is not able to walk on the chromosome to mediate this H2AX phosphorylation. Oh, I didn't put my, okay, so it's very short, but, what we, so from this data, our speculation was that then gamma H2AX spread within a 3D conformation. So we decided to go for IC experiment before and after break induction. And that's basically what we saw. So here you have a differential heat map between before and after break induction, centered on all the DSB we have on the genome. And we could see that we can detect a bidirectional loop extrusion from, so that cross from the DSB. And I'm not showing you the data, apparently, because it's straight to the model. But what we figured out is that that loop extrusion actually helps to establish gamma H2AX. So that's our current model. So basically, coenzyme keeps on extruding loop on the genome. Then what we believe is that it's going to block at DSB where the ATM kinase is recruited, okay? And the ATM kinase will be able to phosphorylate, loop extrusion will still go on one side, and the ATM kinase will be able to phosphorylate the H2AX containing nucleosome as long as the loop is traveling close by to the break. So here I put a ring, but we know it's not necessarily a ring. This occurs on both sides of the break, so that you can end up with this bidirectional spreading of gamma H2AX, and it will naturally stop at boundary elements such as CTCF boundaries. And the main advantage of that mechanism is that in no time, so at the speed of the loop extrusion, which is extremely high, you will be able to go from no histone modification, no H2AX phosphorylation, to an entire TAD that is covered with that specific histone mark, phospho-H2AX, that will tag, if you want, the damaged TAD, okay? And that entire process takes less than 10 minutes. Okay, so now I'm going to spend a little bit more time on that other story, which was about DNA double-strand break clustering. Okay, so we actually, to determine whether we were able to see DNA double-strand break clustering in our cells, we decided to build a cell line that is called, that expressed 53BP1 tagged with GFP. And we could see that indeed 53BP1 foci appear, but let me put that movie. What we could see quite often was that these two independent 53BP1 foci can actually merge together in the nucleus. And that was done using a spinning disc confocal microscope, so we know it's not two DSBs crossing each other. So we decided to use Capture IC at that time, because it was less expensive, and we teamed up with a lab of Peter Fraser to perform Capture IC using 5,000 probes that were established on, that were designed on all the best gamma H2AX domain induced in our system. And so here we could see, so here it's a damaged versus undamaged heat map, and each of the square is 1 megabase of around the DSB, okay? And what we could see straight from this heat map was that some DSBs indeed are able to cluster, while it is not the case for some others. So you take the same heat map, and you just reshuffle it based on the gamma-H2AX signal that accumulate around each of these DSBs, and that's the picture you have. So what it tells you is that gamma-H2AX, the more you have gamma-H2AX, more your cluster, you're able to engage DSB clustering. And more interestingly, what we could see, so I told you we have DSB induced in inactive loci, we have DSB induced in active loci, and what we could see is that the DSB that are able to cluster together are the ones that are basically occurring in transcribed loci and not the other one. So it's specific to DNA double-strand break induced in transcriptionally active loci. So then, more recently, we could perform IC experiment, full IC experiment, and we could reproduce again this DSB clustering. So here again, you have a differential heat map, and you can see this very nice clustering of these two DSB here, these two gamma-H2AX domain. And what is important here with the IC, we could see that it's the entire gamma-H2AX domain that is actually able to cluster and fuse together, okay? So this process is entirely dependent on the ATM kinase, because if you inhibit the ATM kinase that is establishing, that is phosphorylating H2AX, then you completely lose this clustering ability, or strongly impede the clustering. And this is not dependent on DNA PK inhibition, which is another DNA damage responsive kinase. Okay. And if anything, it rather increases the DSB clustering. Okay, so until now I've talked about DSB clustering. So the idea was, can we actually use what is classically used to analyze IC experiment and to detect chromatin chromosome compartment? So again, I mean, this has been introduced many times at this meeting. The chromosome make this B compartment, which is basically heterochromatin, and the A compartment, that is the rest of chromatin, and that you can actually determine by doing some principal component analysis on your IC maps. So we decided to do this PCA, but rather than using individual heat map, we used the differential matrix between after damage compared to before damage, in order to really only look at what is changing and not the behavior of the A and the B compartment, okay? And when we did that, so here you have the eigenvector of the first principal component of this PCA analysis. And what we could see is that we have these peaks that coincide very nicely with the gamma-H2AX pattern. So this is an IC experiment, okay? That's the gamma-H2AX profile. And so we decided to call that the D compartment for DSB-induced compartment, basically, which tells you that this cluster with that, okay? So form a specific compartment in the nucleus. But the true surprise arrived when we actually figured out that some loci were able to be encompassed in this D compartment, so basically cluster with the gamma-H2AX foci in vivo, but did not display some gamma-H2AX signal and do not display any DSBs, actually. So undamaged loci were also able to cluster with and to form this D compartment and to self-segregate with the DNA damage themselves. So we retrieved all these loci, and we could see that basically they rather carry active mark and are rather devoid of heterochromatin mark. So somehow it coincided, those places, those genomic loci were active loci, genes. And so to cut a long story short, I'm just showing you here an example. So we figured out that those places that go to the D compartment are actually genes from the DNA damage response. So here I'm showing you an example. Here you have 3 independent replicate IC experiments, one that was performed with a DNA PK inhibition, okay, as well. you have this D compartment here that is very clear, coincide with GAMH2X, and you have this locus here that is reproducibly enriched in the D compartment, no matter the experiment. And when you zoom in, we could see that it coincides with the localization of that very famous DNA damage responsive gene, which is GAD45A, which is strongly activated post-DNA damage. Okay, we could validate that by performing RNA-scope experiments. So basically, we identified genes that are DNA damage responsive, so activated post-DSBs, and that co-localize with GAMH2X loci, with the DSB themselves in the nucleus. And this is not the case for, so we could see that indeed some gene like PLK3, that is a DNA damage responsive gene, co-localize with GAMH2X, and that's not the case for some other genes, including some genes that are also activated by the DNA damage response. So it's not like all the genes from the DNA damage response goes to the D compartment. So what's the point of targeting some gene physically in that D compartment? Well, it contributes to their optimal activation post-DNA damage. So we use the trick here that you may understand a little bit later. So we knock down the SUN2 experiment because we know it's involved in DSB clustering, and in no other, to our knowledge, in no other repair events. And we looked at the activation of several DNA damage responsive genes that we know are targeted to the D compartment, and we could see that when you knock down SUN2, then you decrease the activation of those DNA damage responsive genes. So DNA damage responsive genes, so that provide a function, if you want, to the DSB clustering. That was one of the main questions, why the hell would DSB cluster together? Okay, so what we think is that it contributes to optimally activate the DNA damage response and the checkpoint. But it comes with some problem. It has a drawback, which is it promotes translocation. So we have an assay to measure the frequency of illegitimate rejoining between two individual DSBs. And we could see that, for instance, I told you when we treat with DNA PK inhibition, we increase the clustering. Well, we also increase the translocation frequencies. And when we decrease the clustering, which is the case when we do siRNA against 53BP1, SUN2, ARP2, well, we did that as well, but I'm not commenting too much that. Then we could see that we decrease the translocation frequency in the condition where we decrease the clustering. So that's currently our working model for that. So what we think is that once GAMH2X has been established, this GAMH2X domain, they can actually self-segregate. If you leave enough time, they will start to self-segregate and to form another DNA double-strand break induced chromatin compartment. This coincides with delayed repair. This is mostly for DSB induced in active loci. What I have not showed you is that it somehow seems to depend on the accumulation of R-loop. If you so those genes, they are an R-loop rich. If you work with in absence of senataxin that help to resolve R-loop, then you increase the D compartment formation. So we have several lines of evidence that depends on R-loop formation. And what is the most important is that it promotes the activation of some DNA damage responsive genes. So while promoting translocation, it still has a benefit for the cell since it helped to activate the DNA. It fine-tuned the DNA damage response. Okay, if I still have a few minutes, Angela, what? Good. So just for the rest of the talk, I would like to discuss with you a new story we have, which is about the relocalization of the DSBs. So until now, what we knew is that persistent DSB needs can relocate to the nuclear envelope, to the nuclear periphery at different points on the nuclear periphery. Okay, that's also the case for DSB induced in heterochromatin in Drosophila. Okay, and that's also the case for arrested fork in yeast. But we didn't know yet whether DSBs induced on the human genome could actually display that behavior. So we decided to, I just want to show you a movie where we have the nuclear envelope that is targeted, that is labeled with lamine B1 in cherry here, and 53BP1. And so some of the foci are already at the nuclear envelope, as you can see here. But if you follow this one here, I hope you will see, then it's pulled to the nuclear envelope quite rapidly, actually. So it hangs around, it's already formed, and then at one point it's pulled to the nuclear envelope. So we perform chromatin IP experiments against lamine B1, and we could see that indeed lamine B1 is recruited at DSBs that are induced on transcribed loci. So what we call the TC-DSB, DSB induced in transcriptionally active loci, but not on DSB that are induced in intergenic loci. Okay. So we have other piece of evidence such as PLA, but then we were wondering about those SUN protein that are part of the link complex. So for instance, I mean, we have many evidence, so in yeast, in Drosophila, that this link complex and SUN domain protein are actually involved in anchoring DSBs at the nuclear envelope. So we performed cheap experiment against SUN1 or SUN2, which are two paralog to SUN domain protein, and we could see that SUN1 is recruited at DSBs quite nicely, but SUN2, if anything, rather decrease at DSBs. So we performed ChIP-seq against those two subunits, and again we could see that SUN1 is recruited at DSBs, while this doesn't seem to be the case for SUN2. And here you have another DSB induced on a gene that is inactive, and we could see neither SUN1 nor SUN2. Okay, so we have SUN1 accumulation at DSBs induced in transcribed loci. And here you have an average profile of at all our DSB of SUN1, and SUN2 is, if anything, rather decreased at DSBs. Okay, so we wondered whether SUN1 could be one of the anchoring points of DSB in the nuclear envelope, and it seems to be the case because when we deplete SUN1, and we perform a ChIP against lamine B1 at DSBs, then we could not see the enrichment of lamine B at DSBs, showing that it actually triggered a decrease of the DSB anchoring at nuclear envelope. Okay, so then we got some hints from an independent project that were aiming at identifying new factors, new player in this TC-DSB repair pathway, which is the DSB repair pathway that allow to repair break induced in transcriptionally active loci. Okay, so we know those DSBs need specific repair pathway, need specific actors. So we decided to start a screen to find more of these actors, and we ended up, so I'm not going to go into this screen, we screened for gamma-H2X foci, translocation, cell survival, and DSB clustering, and we could actually identify multiple proteins from this complex that is called the pair complex, which is a key module of the circadian rhythm oscillation. Okay, so that complex is a transcriptional repressor that will come and inactivate a number of circadian regulated genes, and that has a 24-hour periodicity. And what is important for our purpose here is that this complex also works by targeting the target genes to the nuclear envelope to ensure their proper repression. So, of course, we wondered whether that complex wouldn't be involved also in targeting the DSBs at the nuclear envelope. And indeed, it is the case. So we perform a ChIP against lamine B1 upon depletion of this pair protein. So we have plenty of other things, but I want to show you that. So when you deplete pair 2, then you don't see the accumulation of lamin B at DNA at DNA double-strand breaks. Okay, and so we perform IC, and we looked also at the behavior of that D compartment if you kill the interaction of the DSB with the nuclear envelope. And to our surprise, what is actually happening is that you increase the formation of that D compartment. So I'm showing you here an example. You have the D compartment. This is on a piece of chromosome where usually we don't see that D compartment, which is the chromosome 2 P arm. Okay, so in normal condition we don't see this D compartment, but now when you knock down the pair 2 protein, then we could start to see some D compartment formation that coincide with gamma H2X. So so let me just summarize what I have told you. And what is our actual model. So what we think is that following DNA double-strand break induction, there is a number of chromosome architectural changes that occur. So first of all, cohesin-mediated loop extrusion will take place on both sides of the DSB. Okay? This will allow to establish really rapidly a new histone modification on an entire TAD. So to tag, really, to label, paint an entire damaged TAD. Okay, this has the advantage of signaling the DSB to the cell, forming a DNA damage response focus, DNA damage repair focus, but it also comes with some drawback, which is it can increase the translocation frequency in cis, so intrachromosomal, and this we have shown as well using some translocation assay genome-wide. Then, if this signal persists, meaning the break is not repaired, then this gamma H2X foci that have been, this gamma H2X covered TAD that have been established will start to self-segregate a little bit like would heterochromatin do. Okay, and this is going to form some chromosome, some chromatin compartment that we call this D compartment, in which some genes, DNA damage responsive gene, will also be targeted in order to be properly activated. So again, it has a function, it helps optimal activation, but it also comes with the risk of increasing translocation frequencies. And what we think is that it's very similar to heterochromatin because we think there is a kind of competition between the targeting of the DSB to the nuclear envelope and the fact that it clusters within a D compartment. So basically, if you detach, so the targeting, we believe, help the completion of homologous recombination as it does in Drosophila. And you have a kind of balance between the targeting to the nuclear envelope and the formation of this D compartment, as does heterochromatin when you detach it from the nuclear envelope. And we think that this is going to balance between the repair pathway, homologous recombination, for instance, and the activation of the DNA damage response. So it will fine-tune the DNA damage response. And I also encourage you to go and see the paper from the Huertas lab, who also determined a role of the circadian clock in the DNA damage response, and the work from the lab of Karim Mehail, who also found some interaction of DSB with nuclear envelope structures. And with this, I will thank all the people who did the work in the lab. So the circadian clock story is Nadine and Benjamin, and then the DSB clustering and the loop extrusion was Colleen. Arnaud helped with Vincent Rocher and Sarah Collins for the bioinformatic analysis, and all our fantastic collaborators, Fabian Erdel, Jim Haber, Martin with Aldo, and Dan Nordemer for all the different things. Thank you.
Okay, so first I would like to thank the organizer for this very nice meeting and for the invitation. So I work in the Laboratory of Computational and Quantitative Biology in Paris, and I'm going to talk to you today about multiscale changes in chromatin dynamics upon DNA damage. Okay, so in the team we are interested to understand the physical mechanism governing nuclear organization and nuclear dynamics. Is it better? Okay, sorry. So I'll start. So in the team, we are interested to understand the physical mechanism governing nuclear dynamics and how this dynamics is altered upon different kinds of stress. And to address this question, we use mainly high-resolution imaging, but also genetics and macrofluidics, both in yeast and in human cells. So today I'm going to focus on the changes in chromatin dynamic upon DNA damage. So about 15 years ago, several labs have shown that DNA damage induces increased chromatin mobility. And to summarize the different results, first, we and other groups showed that in yeast, when there is just a single double-strand breaks, the damaged locus becomes much more mobile, as illustrated here, and it can even explore a nuclear volume about 10 times larger. But that's not all. In diploid yeast, surprisingly, we also found that even undamaged loci are affected. They become more dynamic, creating what we call the genomic-wide increased mobility. In response to multiple damages, it has been also observed both in yeast and in mammalian cells that repair foci can cluster. And finally, in Drosophila, damaged DNA in heterochromatin can actively move to euchromatin to get repaired. So these are more experimental results. Okay, sorry about that. So I was about to say that a lot of this work has been done in yeast, as illustrated here. We can see the trajectories of chromatin loci here, and when there is no damage, you can see that they explore a small nuclear volume, and then when there is a single double-strand breaks, you can see the difference, and when there are multiple double-strand breaks, you see a very large difference. Many of these observations have been also observed across many different organisms, such as Drosophila and human cells. And it doesn't move on. Okay. Yes. Okay. And just to give you the most impressive example, the differences in dynamic is very dramatic. So you can really see here when there is no break and when there are multiple breaks, how two chromatin loci can really move differently. So what is happening exactly? It has been proposed that these changes are due to intrinsic chromatin modifications. So indeed, some labs have shown that upon DNA damage, chromatin becomes stiffer due to phosphorylation or also due to later the repair proteins RAD51. Another observation in haploid yeast from the Gaelle lab is also that there is an important loss of histones inducing chromatin relaxation. On the other hand, several studies have proposed that chromatin becomes more mobile because of external mechanical constraints. So, for example, it has been shown that some physical constraints are released at the SPB and at the telomere in budding yeast also. And more generally, there are also a very important emerging roles of the cytoskeleton, which has been shown in yeast, Drosophila, and in human cells. So there are many open questions. So for example, one important caveat of this experiment is the fact that we induce most of the time breaks in bulk, and when we look at them on the microscope, they all had a break at a different time. So the precise timing of these changes in chromatin dynamic is not understood. Probably there are different mechanisms that can trigger changes at different times after breaks. And finally, we want to understand what are these changes important for the proper DNA repair. So we come back to the basis. To understand better what happens, we really start by the very, very first event happening after damage. And this event is the recruitment of PARP1.
So here you can see the PARP1 proteins in live cells, in human cells, and what it does is that PARP1 is recruited here on this line, which is irradiated using UV, and PARP1 will create PAR chains of about 200 ATP ribosome units. It will pyrylate PARP1 itself and also histones. And as you can see here, there is a peak of PARP1 recruitment 30 seconds after the damage, so it's very rapid. And then it slowly disappears, and allowing the recruitment of other repair factor. So let's focus on this very early event. So what does it do exactly, PARP1 and histone? And it has been shown that histone spirulation triggers transient chromatin relaxation. In fact, in vitro, we can really see that histones are less spaced. And in vivo, it has been shown that chromatin decondensed due to PARP1 pyrylation. So to understand this chromatin remodeling event, we use macroirradiations to measure compaction at the lesion, to measure the mobility of a single chromatin locus, and also to measure the individual histones mobility, all in human cells. So in the next slides, I will always refer to the three scales that I described: nuclear scales, the fiber, and the nucleosome. But first, let's see what happens at a large scale. So in that case, if we irradiate a cell, here we manage to observe by fluorescence only the damaged area in live cells and to measure the decompaction of chromatin. What we observed is that chromatin indeed undergoes a transient unfolding upon DNA damage. Then let's zoom in on the fiber scale. And here what we see is that if we measure the dynamic of a specific chromatin locus, it exhibits anomalous motion in the absence of break, here in blue. But then if we irradiate close to this tagged chromatin locus, the behavior is dramatically different. In fact, we see that the MSD follows almost Brownian motion, and at a larger time scales, it's even super diffusive. Okay, and then if we irradiate far from this tagged locus, it's quite different. There is a slight difference, but nothing compared to the damaged area. And now let's move on to the smaller scale, the nucleosome scale here. So what we do is that we perform single particle tracking of histones in live cells after irradiation. So the rectangle here shows the irradiation area before damage and 1 minute after damage. We showed here 1 minute because it's where everything happens, but we have done it at many different time points after damage. So as you can see here, we plotted a diffusion map of the histones H2B with a color. And if we zoom, we can really see that the types of trajectory is changed. So to quantify this, we have classified the different trajectories in 3 different classes: the slow, the mobile, and the hybrid. So what we saw is that the slow histones are the most abundant, of course, and upon DNA damage, there is a slight decrease of the slow population. But when we look at the other population, it slightly increases. And we also saw that the mobility, the global mobility of histone increases 1 minute after the irradiation. So, what is happening exactly? So, is there a role for PARP1? We saw that PARP1, in fact, is a key regulator of chromatin breathing upon damage. So we looked again at these 3 scales, and what we found is that if we use an inhibitors of PARP1, we used talazoparib, we couldn't find again this chromatin expansion following DNA damage. Instead, we found that chromatin is overcompacted after PARP treatment, and at the scale of the LACO, the mobility is even decreased instead of being increased, as we have here in the control. And then to look at histones mobility, we plotted for each trajectory the mean distance covered in 10 milliseconds in 1 delta t. And when there is no PARP, after 1 minute it's much more mobile, but when there is a PARP inhibitors, histones are much less mobile. So PARP1 is a key regulator of this chromatin breathing upon DNA damage. And this is part of the model we proposed. So these are our 3 scales we are looking at. Then we induce the break. We have parylation here by PARP1. So it creates these chains on histone, as I showed at the beginning. And we have chromatin unfolding, increased chromatin dynamics at the level of a gene, of a LACO, and increased histones mobility, which is really triggered by PARP1 parylation. So to go further, we wanted to know what happens at longer timescales as well. And recently, in the MATIC lab, they have shown that there is deparylation, but it happens in 2 waves, in two steps. First, you cut most of the tree, most of the part chain, so you just end up with this small mono ADPR, and then with another protein is responsible to really cut this last mono mark. And they have done this using specific antibodies against poly-ADPR and then against mono-ADPR. And they show that the kinetics is quite different between deparylation and demarylation. So we call this MAR marks, this one. So we used a mutant in which the MAR marks cannot be removed, so they are always here. And in that case, we showed that at the nuclear scale, if we induce a break, chromatin will expand because you have the parylation, but it will stay open for a very long time if we don't remove the MAR marks. And same thing at the nucleosome scale, we measured again the mobility of individual histones, and 1 minute after break they move more, but we found that 10 minutes after break they move even more instead of going back to the initial level. So we proposed a model in which we observed a multiscale chromatin breathing upon DNA damage. So first, when we induce the break, we have histone parylation triggering chromatin unfolding and increased chromatin dynamic. But then the removal of MAR marks is responsible for the reclosure of chromatin until the initial state, to reach the initial compaction and dynamic state. So the story is not totally finished because, in fact, we also observe that at even later time points, chromatin becomes more compact and less dynamic, but this will be for another story. So I would like to thank all the members of the team. So it's mainly Fabiola Garcia Fernandez, who has a poster here. I would like also to thank June Park, who is doing a lot of image analysis and the classification using deep learning. We also participate to a challenge because we are rewriting tracking algorithm to improve them. I would like to thank also the Taddei lab, where we built the prototype macroscope to combine SPT and macroirradiation, and the Sébastien US lab, who is expert in PARP, and Ivan Matic lab, who is expert in MAR. And also, of course, we are always interested to develop new modules on macroscope in collaboration with the company Aurèle. So thank you very much, and I'm happy to answer to your question.
Thanks, that's very nice. I think in answer to the question that asked me earlier whether we saw histone degradation after damage, and what you've shown, that parylation goes way up and everything's more flexible. Do you think that, I mean, one thing is that the parylation could also serve as sort of a histone, as a way to evict histones. And a long time ago, it was shown that histones would bind pyrrolation. Do you think that's a mechanism, that it's not just opening chromatin like kicking off other factors, but it could actually be mobilizing histones away from the DNA?
I'm not sure.
Fraction.
Okay. If it's the case, it would be a small fraction. I don't think there is a large fraction of histones which are mobilized away due to pyrrolation, because if it was really the case, we couldn't do the experiment, because we really look at single histones, and we can look for 5,000 frames before irradiation and 10 times after the irradiation, and we always use the same photoactivation, so we don't see an important loss of histones.
What about H1?
H1, maybe. I'm just talking about H2B, which we are looking at. It's possible that there is a loss of histone for another one. That could be an interesting point to check.
So
indeed.
actually, I didn't mention it, but in yeast, much more important than the core histones is the H1 homolog. And actually, we did show that if you degrade the H1 homolog, you actually accentuate the recombination. So I think actually probably, yeast doesn't have as much H1, or it's not as important as in mammalian cells. But I'm still wondering if pyrrolation doesn't maybe mobilize some histone, maybe a certain variant.
So this is a good point, but I think in the Selou paper from 2017, they showed that H1 is sliding, it's slightly more mobile, but they haven't used a SPT, so they had this idea that maybe for H1 it has some role. I don't think 30% is evicted or something so strong. It's really lighter.
Hey, Judith here. Very nice. So you showed dynamics up to 10 minutes, right? 600 seconds here in your. So is there a difference in dynamics on the three scales depending on how far they are from the nuclear envelope? Because we heard that the nuclear envelope is an important component of DNA, DDR. It has things have to go to the envelope, etc. So is there a difference?
It's possible, but we will need to look at. At the scale of a LACO, we will need to use different cell line where we tag other sites. At the scale of the histone, in fact, the difference in mobility is really local at the irradiated zone, and we have tried only to irradiate something around the middle of the nucleus. So we could do it. It's something that we need to investigate. Irradiate on the side and see if there is a difference for histone's mobility or even chromatin expansion.
Right, and this is just a question to clarify. So in the last talk we heard there is local changes, then there is clustering, and then things move to the periphery. What are the time scales in general of this whole process? From initiation to clustering to movement to the nuclear envelope.
At the moment here we focus on the first 15 minutes after the break, and I'm sure what Gaelle was talking about was at a much longer time scale. I think what Gaelle was talking about happens at a much longer time scale than what we are looking at, because here we really focus on the early chromatin event happening between like 30 seconds after the break to 15 minutes. But the story is not finished. We would like, for other stories to continue. We have solved some technical problems to really do this kind of experiment even 30 minutes after the break or 45 minutes. And
how much time does it take to repair, more or less completely, and if that's possible?
How long time does it take to form the D compartment, for example? That's I don't know exactly. I would say more than 30 minutes. 30 minutes, an hour, 2 hours. I'm not sure exactly. Okay. If Gaelle is here, maybe she knows. But I'm not sure it's so easy to quantify because we are doing microscopy with a precise time zero for the break. It's quite different in other systems to have this to feel these timings.
Thank you.
Judith. Yeah. So you showed this image where, the graph where you treat with PARP inhibitor and you induce the break, and what you see is that actually chromatin compact if you inhibit PARP1. So can you comment on this?
The fact that it is even less mobile when we have PARP inhibitors.
Yeah, and that makes
this is an interesting point, and it's also something we will follow up. When we have PARP inhibitors, they do something called PARP trapping. So the PARP proteins, they are still there. It's not a KO. The PARP1 is there. It's go on the break, and it stays there. So it's trapped, and probably the PARP1 trapping it has consequences on chromatin mobility, and what we see probably is due to that.
Judith, very nice story. I wonder what might be the role of this, because 1 minute is a pretty short time, and everything else takes many minutes after that. What might be the role of this PARP relation? To keep the two ends together? Is it possible to track the two ends?
So I have a slide on that. So it's interesting for physicists to look at chromatin mobility, but is it exciting for the biologist? Is there consequences on DNA repair? And we think when there is not chromatin reclosure, as we see due to the demyelination, it will change the kinetics of the protein repair recruitment. So what we show is that here we look at 53BP1 recruitment in living cells, in live cells, in this mutant that do not do the demyelination. And we can see that it boosts 53BP1 recruitments. We don't see an effect for BRCA1, but we have looked only at 2 repair proteins at the moment. But the idea is that probably this chromatin breathing and then reclosure, or even overcompaction after, it has a role, and it will modulate the recruitment of repair proteins. So it has an impact on other further steps.
Can it be important to keep the two ends of the break together?
But the two ends of the break are kept together due to proteins. It's in fact, there are proteins that have a role to keep the two ends together. And this comes back to Vittorio's question. If you have a break, like it's true that the physicists, they see that as a release of constraint with the two breaks going far away, but there are proteins that keep the ends together, and you have to see that as, okay, you break something, but you put some anchors, and it changes the flexibility, but maybe it's good do that also.
Are proteins recruited without PARP-relation?
Which protein is recruited if you don't have?
It does not need correlation.
It does not?
No. Yeast has no correlation anyway.
Yeah, yes, that's true.
So much of the first MRX, MRN, and CU, and ATR.
I would like to ask you something.
Yes.
With what did you label H2B? With what fluorescent protein?
For the single particle tracking, we use H2B halo, and then we put Halotech GF photoactivated versions in the cells to visualize them.
But in this case, when you microirradiate, you activate actually. You photoactivate the...
Very slightly, it's true, but we are using a very low macroirradiation compared to most of the literature, especially because we are doing single particle tracking. In fact, when we built the setup, we had to put the lowest laser we could buy in terms of power. We had to add density optical density, otherwise it was just exploding the cell in terms of fluorescence, because we have photoactivated fluorophores. So we are quite low. We use, I think, the lowest power for the macroirradiations to be able to see PARP1 recruitment without photoactivating too much the tagged histones.
But with what I see, when you cut and recruit part 1, you can see a lot of bleaching actually.
A lot of bleaching? A lot
bleaching.
Of bleaching. Yes, but it's really, then it's true that when we do the experiment, we cannot do SPT for 15 seconds, for 10 seconds, let's say. This is what the first time point we have is at 15 seconds after we irradiate. But so far, this is the best we can do.
Okay, thank you. Thank you.
But it's a good point, it's true.
Okay, no more questions for Judith, so let's thank Judith again.
All right, so thank you a lot to the organizer for inviting me to give this talk. It's not so often that a young faculty like me gets invited yet. So what I'm going to show you today is something we initiated in my laboratory, which aims at better defining the mechanism of homology search during the repair of DNA break during homologous recombination. Okay, so it's basically it's in the field of target search, but it's not like classically studied in the field of transcription factor, multiple transcription factor looking for a target in DNA. It's a very particular case of target search in which both the search entity, the break site, and its target, the donor site, are both part of the DNA polymer, which means that they have subdiffusive behavior, okay? And there are only one searching entity and one target site. So it's really a main question in the field, how this extreme case of target search happens. And you can really break it down in two main questions. The first aspect, the special aspect, is how can you provide opportunities for special collision between two subdiffusive pieces of DNA that have to find each other. And the second one is how the identity between the broken molecule and its donor can be identified. So essentially it's more like a molecular aspect of the search, which is how homology is being recognized in a massive excess of heterologous DNA in the genome. So going down a little bit into the complexity of the pathway. So once a break is formed, it's resected. So it generates single-strand DNA. And this single-strand DNA is used as a platform to assemble a filament of the Rad51 recombinase. And so this is a search engine that we are going to talk about. And it assembles on both ends. And then there is a series of steps where essentially this entity has to engage with the DNA that collides with it in space and sample it for the presence of homology until a homologous donor is identified, which will lead to the formation of DNA joint molecules from which DNA synthesis can be primed that essentially restores the sequence information that was disrupted at the break site. And then I'm not going to go into this, but final steps of the pathway lead to either non-crossover, conservative repair outcome, or crossover. So the first step, as you can see, are covalent alteration of the substrate, and the last step are covalent products, and those are easy to detect molecularly, but all the core steps of the pathways are just transient intermediates that are actually prove extremely difficult to detect. And so worked in my postdoc aimed at finding ways to detect these DNA joint molecules, but what I'm going to talk about today is a method to detect what's being actually sampled over the course of homology search by this Rad51 filament. Okay, so that's the main question. I'm going to describe this methodology, which is based on IC, and what we learn from it. So the system is in Saccharomyces cerevisiae. We have a very nice system where we can make a single break at a defined position extremely efficiently in 30 minutes, more than 90% of the cell have the break. And we can provide a donor, which actually corresponds in this system to only one side of the break. Okay, and you will see why in a minute. So we used this system in the past with IC to address and to answer the question, what happens to chromatin
in 3D when you make a break, and how this organization influences the downstream repair steps. Okay, and so once you make a break, a number of features happen along the main diagonal of here the broken chromosome, chromosome 5, which is true also for all the other chromosomes, which is the appearance of loops that are mediated by coenzyme. And at the break level, you see this tethering, this fountain that connects the left and right end of the break, and that we showed, and I'm just going to summarize here with a model, depends on the 911 clamp and long-range resection by the, actually short-range and long-range resection factors. So this blocks coenzyme. So coenzyme accumulate in the vicinity and extrude unidirectionally from the break site, which essentially position this resection front at the axis of metaphase-arrested chromosomes. Okay, so at the base of the loop, regions that are point of contact between sister chromatids. All right, so there is one thing though, is that IC only detects double-strand DNA contact, and so we were essentially blind to anything that the single-strand DNA that is generated by resection actually can contact and see. So to overcome this limitation, so this is a classical IC, right? You have two pieces of double-strand DNA held together by a cross-link, you digest, biotinylate, ligate, and then you can recover the contact. So obviously this does not work if you have single-strand DNA bound to double-strand DNA, because the restriction site is ablated in single-strand DNA. So what we did was to restore this restriction site by, at the lysis step, including oligonucleotides that will be able to anneal on this single-strand DNA, and those oligonucleotides contain a few polymorphisms that will allow to distinguish afterwards which contact actually came from single-strand DNA, from that site in the single-strand DNA state. So then we can get our IC library. Among the contact we detect, there is the site of interest in a double-strand DNA form, the site of interest in a single-strand DNA form, and many other pairwise interaction classical of IC. So it's really a tiny fraction of the molecule. So what we do is then we use targeted capture with biotinylated oligo against the site of interest to enrich it. And then we use a trick is that actually the SNP we introduced inactivate a secondary restriction site that allows us specifically to digest the contact made at that site in the double-strand DNA form. So it isolates contacts made at that site in a single-strand DNA form. And we still have a bunch of pairwise contact that come from other generic regions. So we added oligonucleotides to restore site at various distances from the break site, up to almost 20 kilobase, to cover the range of resection that is achieved within a few hours in Saccharomyces cerevisiae. And so when, and so here I'm showing you the data of a regular IC experiment and what we call the single-strand IC experiment, and that you can see that single-strand DNA IC recapitulates really well everything we see with a IC experiment, but that in addition, at the break site, reveal additional contact that were not visible in the classical IC experiment. It reveals the contact that are made by the single-strand DNA, and that, as you can already appreciate from this map, are much less, are capable of engaging other chromosomes, okay, making contact in trans. So we can represent this as a 4C-like representation. Here in red, I show the profile of contact of the double-strand DNA. So this is IC double-strand DNA on the left of the break, blue is the right of the break, and black is a control site that is at the same distance than the break site from the centromere, okay? And then single-strand DNA is the same, except that here we are looking at single-strand DNA contact. And immediately what you can see is that the contact made by single-strand DNA are much less constrained in cis and are much more distributed on other chromosomes than contact that are made by double-strand regions. Okay. The other thing is that you see the distribution is uneven, so there are peaks that correspond to centromeric regions. So the break site is close to a centromere in our system, so that reflects essentially that proximity between the centromere and the yeast genome. But also you can see other sites, so there is a peak at the donor, and there is an enrichment specifically on this chromosome 3. Okay, so I'm going to talk about the specific sites, but first I want to give you, a sort of a general overview of the contact distribution made by single-strand DNA. So if we decompose the fraction of contact that are made at the different sites that we have restored on the left and right of the break, what we can see is that the single-strand DNA sites that are close to the break site are making way more contact with other chromosomes than the single-strand DNA sites that are closer to the resection edge. Okay, so this means, and if we look at the distribution of contact that those sites make with the chromatin that is flanking it, we see that sites that are close to the break in red are making much more long-range contact than sites that are close to the resection front that tend to fit with the genome average contact probability as a function of the genomic distance. Right, so this tells us that there is a special disconnect between the break proximal single-strand DNA site and break distal or close to the resection front single-strand DNA site. So they are not at the same place in space. Okay. So we teamed up with Daniel Jost, our neighbor at the ENS, and he came up with a simple Gaussian polymer model of single-strand DNA where he has a stochastic description of resection, and from there can, with varying the persistent length of the single-strand DNA chain, can output the expected fraction of trans contact and the distribution of contact in cis with chromatin. And so we ran just varied this parameter, and you can see that with a persistent length of single-strand DNA of roughly 20 nanometer, you can pretty well recapitulate the trans contact as well as the distribution of contact in cis that we observed experimentally. So this is obviously a measure that corresponds to a whole population of cells where single-strand DNA can be in different states, not necessarily always stretched. But it means that you can explain these profiles simply by the rigidity of the single-strand DNA that is being generated. So what provides this rigidity? So obviously, the candidate is Rad51. Okay, Rad51 makes filament, and it's known for a long time that the persistent length of Rad51 filament is a conserved feature of this protein, and that makes that approaches in bacteria 1 micron. So if you remove Rad51, this is the left end of the break, this is the right end of the break, this is the control. You can see that you lose the trans contact. So all the contact at a distance are dependent on Rad51. If you look at the distribution of contact in cis as well, so the long-range cis contact are dependent on Rad51. So the rigidification of single-strand DNA is provided by Rad51. And if you don't have Rad51, you don't have naked DNA, but you have RPA-coated single-strand DNA, and this clumps and essentially has a very small persistent length. So the question was then, okay, so resection generate long single-strand DNA, and is this required for the assembly of those long filament? So we turn to EXO1 mutant that is defective in long-range resection, as you can see on the high-C coverage profile. And what you can see is that the distribution of trans contact is also strongly reduced. So long-range resection in the field has been always sort of a question, is that why do you need to generate so much single-strand DNA? But this shows that it actually required to assemble at least one long filament of Rad51 that is going to be able to look long-range. So that essentially is the same thing in cis. Right? So short resection tract is going to only give rise to short Rad51 segments that are incompetent for reaching at long range, and long resection tract authorize the assembly of those long filament. So what happens to the contacts that are really made close to the resection front? So here I'm just showing the example of 3 single-strand DNA sites. So the break site is here at 0. You have a site that is relatively close by, and you have one site that is going to be, on average, very close to where the resection machinery is. And so we ask, how is chromatin architecture that we know is dictated by coenzyme close to the break site is going to affect the contacts that are made by this nascent site, the site that just been generated by resection. So what we see, so we depleted SCC1 in blue, and this is a wild type, and so you can clearly see that the site that is very close to the resection front is strongly affected by the depletion of coenzyme. And in fact, what you lose is the enriched contact that are present at the site where coenzyme is enriched. So the sites that are closest to the resection, in fact, what they start looking for is whatever is close to the loop base, which we cannot see the sister chromatin, but it's just one safe assumption to say you start looking at the sister chromatin. Then, when you get farther from the resection front, you still are under influence of the coenzyme-mediated chromatin organization, but then you contact mostly the inner part of loops, and when you're even further away, actually, whether or not you have coenzyme to fold your nearby chromatin doesn't matter because you're already so far away, okay? So that's a model. Coenzyme actually exert an effect in targeting the search in the flanking chromatin, but only for sites that are newly generated by resection. So the initial stage is going to be under this influence, but the rigidification of single-strand DNA is going to emancipate the search from that constraint, okay? So this is a first summary about the special features of homology search we could define from this experiment. Is that the first is that homology search follows an expansion program. It starts local, but then the oligomerization properties of Rad51 and the rigidity of the filament over time, in the event of the absence of repair, is going to push the search at greater distances. This is what I just said, and long-range resection is the engine that allows this transition to happen, because if you cannot resect long-range, you cannot assemble long filament. So this really nicely parallel recent studies by Angela Taddei that use functional Rad51 GFP tagged protein, and that could visualize actually those elongated structure, Rad51 structure in cells that accumulate over time. All right, so I told you that there was also a few sites that were enriched for contact that actually we could not detect from double-strand DNA alone, and one is chromosome 3. So chromosome 3 is a bit special because it contains a mating type switching system. So what defines the mating type of the yeast, and that the switch is based on a recombination process. So there is a massive peak of contact at the level of one genetic element that has been defined already a long time ago, called the recombination enhancer, that focuses the contact at its level. And it's really a sort of an early event also. This is completely dependent on Rad51. So if you don't have Rad51, the single-strand DNA doesn't see the recombination enhancer. And this is obviously dependent on this recombination enhancer, as if you knock it out, the chromosome 3 is not contacted more than any other chromosome. So to verify that the recombination was sufficient outside of the context of chromosome 3, we just inserted it in the middle of the long arm of chromosome 4, and you can see that it is sufficient to attract the filament. So it means that in the genome there is elements that are capable of engaging and focusing homology search at certain sites. And the recombination is one very clear example, but I think it's interesting that it's mediated by this function, and from the literature we know this is due to a pioneer transcription factor called FKH1 that mediates this function, and FKH1 binds throughout the genome. So it actually might be an extreme case of something that is going on chromatin, where homology search is helped by a certain factor that might help to recruit the filament. Okay, so one thing that was interesting is that all sites on single-strand DNA see equally well chromosome 3, but the double-strand DNA region that is nearby does not. So that suggests that, in fact, the mode of interaction of the filament with the RE is in an extended configuration. It can happen anywhere along the length of single-strand DNA, but that in general, always, on average, put the double-strand DNA region at a distance. And you can see it a little bit like the archer on a violin. Okay. So the other site that makes enriched contact with single-strand DNA is, unsurprisingly, the donor site, okay? And so I told you at the beginning that we provided homology to the left end of the break, not the right end. The right end has no homology with that site, okay? Yet, what you see is that the left and the right end interact with the donor site. So that means, and this is a 51 dependent and donor dependent. And so that means that the two ends search for homology in a coordinated manner. So now if we decompose the contact made with the donor region along the single, at a distance from the break site, you see that there is this slope that is opposite, okay? So it means that, and the donor is right here, okay? So it means that the site that detects the donor, they are here, and on the opposite end, so the filament that does not have homology, are also sites that are close to the break that engage this donor. And so that suggests a configuration for the search in which the two filaments are co-aligned together in a 5' to 3' orientation, okay? But then, when time goes by, you start seeing that in fact sites that now have been generated by resection that are further away are actually engaging this donor site roughly the same way as sites that are very close to the homology region. And so that suggests that you have a disruption of the filament once homology has been identified at one point, and that the whole resection front now recoil close to the break site. So we were curious to know at what stage of the recombination reaction this disruption will take place. And we wanted to know whether it was coupled to the initiation of DNA synthesis, which is a step where you sort of commit to the repair. So what we did was we depleted Pol 3, so the polymerase that is making the extension together or not with a clamp loader. And in fact, it's still a straight line. The slope is the same. So it means that the collapse still had happened in the absence of synthesis. So our best guess is actually that this is at the DNA strand invasion step where you transition from a synaptic complex in which homology has been recognized in the filament to a DNA joint molecule that coincidentally the old filament get disrupted. And that might be useful for genome maintenance because once you have found a donor, if you find another donor somewhere else, it's probably a pretty bad idea to invade two independent sites. And we know from genetic studies that chromosomal rearrangement can happen that way. All right, so this is my second summary. Homology search is coordinated along co-aligned Rad51 filament. There are specific elements on chromatin that interact with this filament and that bias homology search toward the site, and that homology identification inactivates the filament homology search in a site-specific manner. All right, so this is a fancy picture, but that recapitulates everything. But essentially, there are two phases in homology search. One is initially local, but if homology is not found, we believe that long-range resection authorizes the search to just move at longer distance, probably to get the homologous chromosome. And that is driven by long-range resection. What's really interesting, I think, is that it exploits basic properties of the filament, so the rigidity of the filament. And you can actually see the ability to perform homology search as resulting from this basic property and the size of the segments that you can generate. And so the segmentation is regulated by very basic properties that are specific to this recombinase, the nucleation rate and the growth rate. And if you nucleate very infrequently, but the growth is very cooperative, you are going to be able to make very rigid single filament very efficiently, like Recce in bacteria. However, if you nucleate frequently, but the growth is not very cooperative, you will make a lot of small segments. And this is not going to be able to grow like this. So what we start to study now is the maturation of this filament that is probably requiring rounds of disruption and reassembly, at least in eukaryotes. So I'm going to stop here and thank my lab. So most experiments have been done by an engineer in the lab, Agnès Dumont, who also worked a lot to improve and develop the single-strand IC technology. And a lot of the analysis was possible thanks to the development of a pipeline and great help from Nicolas Mandibour, a grad student in the lab, and also our collaborator, Daniel Just. One last slide, because we heard a lot of talk about SMCs and condensing yesterday, and I felt bad because we have these things coming to an end. So it's completely unrelated, but we found that the RE actually, besides attracting the filament, is also a condensing loading site in Saccharomyces cerevisiae, and it makes a very nice stripe. Okay, so this is great because it allows you to study basic properties of loop extrusion by condensing in cells, and so I'm just summarizing here for anybody interested in condensing that wants to talk with me, that it really extrude loops unidirectionally with that processivity. We identified a bunch of fault blocks, some of which were actually pretty unexpected. Top 2 is required for extrusion, but actually it's because it generates substrate that are bound by SMC56, and that is the one thing that blocks extrusion by condensing. It's blocked by forks and by breaks, and it has a function in mating type switching as biasing the search towards the donor that is close to the recombination enhancer. So okay, I'm stopping here, and I'm very happy to take your questions.
Yeah, that's a very nice method. I was kind of blown away by these maps. I'm trying to reconcile two things you said. One was that the model was the single-stranded, that a model that reproduces single-stranded DNA with the persistent lengths of 20 nanometers approximates the data well, but at the same time you say filament is what should have been there instead of the single-stranded DNA that presumably has a persistent length of like hundreds of nanometers.
Yes.
Do these models also? Did you try modeling that?
So a very good point. So you would expect a much greater persistent length, but this is average of a whole cell population. Some cells that might have extended structure, and some cells that maybe, as we know from the dynamics of those filaments, be coiled or not have filaments at all. So if we start taking this into account, we actually get way bigger in the order of 100 nanometers persistent length. Yeah.
Right. And the other question, I was trying to understand the role of cohesion. You're saying, so cohesion, when you get rid of cohesion, you lose some local context.
Yes.
Is this right?
Yeah.
And you say this might be context with the sister. So can you now extend this method such that there is no sister?
So we
sister resolved?
Yeah, we did an experiment in which we prevent replication upon S-phase entry. So you accumulate in metaphase, not metaphase, but chromosome compact, and there is no sister. And no, you still see the peaks at a position where cohesin is enriched. But since position where cohesin is enriched, so the base of the loop are often the position where there is also cohesion, it put them at the same place, location. But yeah, it's a limitation of the system. We have no way to study intersister repair, first because the two sisters are broken in our system. The second one, we cannot detect intersister versus intrachromatid contact. Yeah.
Very interesting talk. Could you go back to the second slide you showed of your previous paper with the contact map of the plain old high C? What you showed there was a very symmetric looking fountain. So I guess which seems to maybe indicate that the reeling in is synchronized across the two sides of the break. So do you have any ideas on how you can actually synchronize that process on both sides? Or maybe I'm not looking at it correctly.
Okay, so this I must say, the perpendicular fountain is not cohesin mediated. Okay, it's cohesin independent. This is the connection between the two resection front. So resection proceeds on double-strand DNA independently, sort of, but they are held together in space by factors. So, okay, this we can discuss. But so this is it results from the probably uncoordinated resection of both breaks. Okay. And we lose this, and in fact, but the problem is that there is no good mutant that allow you to uncouple the resection from the tethering of those two extremities. So if you remove EXO1 or the MRX complex, you essentially lose resection, or have impaired resection, but you also lose a connection between the left and right DSBN. So the chromosome fragment that has no centromere start floating around them. Yeah,
in line with the same question, what is the size of the resected sequence, and how does it relate to the structure that you show here?
Can you repeat, please?
What is the length of the resected sequence,
yes,
and how does it relate to the sequence that you show here, to the
yes,
IC map?
Yeah, so resection proceeds at 4 kilobase per hour. This is 4 hours, so roughly 16 kilobase. And this is what I show here. So the window here is plus minus 20 kb on the inset.
So I have a question, a more general question, but if you look at the persistent length from of a Recce filament, for example, and then they are super stiff, yeast are stiff but a little bit less, the persistent length is a little bit below, and then human cells, it's a little bit below, even more, at least in vitro, with the protein alone.
Yes.
So I was wondering if this is a general, if it's really a general in the evolution that from yeast, from E. coli to human, the filament is less stiff, and or maybe it's because they need partners to be stiffer.
Yeah, so it's a very good question, and there seems to, so as you say, in eukaryotes there is more partners to the filament. Recce is very autonomous in doing everything. So yeah, you can see it as an extra layer of regulation on the structure. You may not want to do a very stiff filament right away or very extended filament right away. So it's possible that there is a sort of a maturation that is implemented through these different factors, and we are having a study, I didn't talk about it, but the RAD51 paradox, that RAD55-57, that associate with the filament, stimulate nucleation, but also protect against dissociation by SRS2, could play a part. So the combined dissociation of small segment and reassembly of filament might allow for the progressive growth of a few selected filament. So yeah, I mean, it's perfectly possible that there is an extra layer of regulation imparted by this associated proteins.
Maybe RAD54 also for to stabilize the RAD51 on the single strand.
So we did 54 actually. I don't think there is single strand DNA data, IC data here, but no, 54 has exactly the same profile of contact as a wild type cell. So it does not change the structure. However, it changes how much DNA is engaged by the filament. And we have also the the ATPase mutant that behave not exactly like deletion though. Yeah.
Thank you.
If I may comment on this, like by microscopy, the filaments are not very different, neither between 54 delta and the wild type. So, I mean, it fits.
My question is, very nice method. How much resection does one actually need to do the search? Because I think it seems to me this is an exceptionally long time, and it's not actually necessarily physiological. Although I'm sure the thing you described is physiological. I mean, the loading. I mean, obviously RAD51 and all that. But what would if you restricted resection to? We know that you can recombine with 40. I mean, 40 nucleotides, or
Exactly.
If you restricted it, what impact would that have on your mapping of peaks? Do you think this would have an effect?
So we need single-strand DNA to detect single-strand DNA contact. If we completely ablate resection, we won't detect anything.
I don't mean ablate. I mean short.
Yeah, but shorten it. So the thing is that already with EXO-1 we have only very few, especially at 2 hours, we can only detect what's going on very, very close to the break site. We didn't try the EXO-1 SGS double mutant to make it super short because we have no site within the 1 kb of the break. So we cannot really just do that experiment. Yeah.
Okay, so it's a detection question, not really.
Yes.
I mean, because.
We did D-loop capture experiment, so we are not looking at exactly what the single-strand DNA sees, but we detect the intermediate of successful homology identification at an interchromosomal donor. So in fact, you manage to have it with the EXO-1 SGS, so you don't need this extra long to find the donor. But I don't remember. I think I need to do a careful time course analysis to see if there is a delay though. Because, so then the question is, why do you need so much single-strand DNA to assemble, to be able to contact in trans like this? I think the answer is that you increase the probability that there will be within this single-strand DNA a long segment formed that is going to be able to separate the sites that are closer to the break from those that are near the resection front. Yeah, just a probability. Yeah.
Okay.
Any other questions? Okay, if not, we should thank Aurèle again.